Welcome back to Edjo Learning Platform. Today we are learning about analytical technique in order to find adulteration in the food. It is very useful for FSO exam and all science related objective exam. Today we will discuss about chromatographic technique, column, paper, TLC, HPLC and gas chromatography. So, first of all, we have to look at the history of chromatography and who is the father of chromatography. Just you look, it's a Russian scientist Mikhail Swift in 1900. He continued to work with chromatography in the first decade of the 20th century, primarily for the separation of plant pigments such as chlorophyll, carotenes, and xanthophyll. Since these components have different colors, green, orange, yellow, respectively, they gave the technique its name, chromatography. Chromatography is literally known as color writing, and this is a scientist named Swift, Russian botanist, is created for the development of chromatography. Then what do you mean by chromatography? That is chromatography. It's a Greek word, come from Greek. Chroma means color and graphene means to write. The collective term for the set of laboratory technique and for the separation of mixture. The mixture is dissolved in a fluid called mobile phase and which carries it through a structure holding another material called the stationary phase. The various constituents of the mixture travel different speed, causing them to separate. The separation is based on differential partitioning between mobile and stationary phases. Chromatography, which contains two phases, one is stationary phase and another one is mobile phase. Chromatography is a physical process where the component of a sample mixture and is separate as a result of their differential distribution between stationary and mobile phase. Greek chroma meaning color and graphene means writing. Just you look at the picture, mobile phase and stationary phase. Mobile phase is usually refers to the mixture of substance to be separated, dissolved in liquid or a gas, whereas stationary phase is a porous solid matrix through which the sample contained in the mobile phase percolates. Chromatography is usually based on principle of partition of solute between two phases. It is usually consists of mobile phase and stationary phase. It's the main principle of chromatography is the partition of solute between two phases. Chromatography usually consists of mobile phase and stationary phase. The mobile phase refers to the mixture of substance to be separated, dissolved in the liquid or gas. Stationary phase is a porous solid matrix through which the sample contained in the mobile phase percolates. The interaction between mobile phase and stationary phase result in the separation of compound from the mixture. So once again, just you note what you mean by mobile phase. Mobile phase refers to the mixture of substance to be separated, dissolved in the liquid or gas. And stationary phase is a porous solid matrix through the sample contained the mobile phase percolates. Once again, we have to know the classification of chromatography. Chromatographic method can be classified into three different ways based on the shape of chromatographic beds. Example, planar and column chromatography, Based on the physical state of mobile and stationary phase, example gas and liquid chromatography. Based on the mechanism of separation, example ion exchange chromatography, partition, affinity and absorption chromatography. So once you have to study in detail the chromatography based on shape of chromatographic bed. Example planar chromatography or column chromatography, and based on the physical state of mobile phase and stationary phase, gas chromatography and liquid chromatography. Chromatography that based on the shape of chromatographic bed is again divided into planar and column. This planar is again divided into paper and thin layer chromatography. And next one, the column. Column is again divided into gas and liquid chromatography. So we have to look or we have to study more about planar chromatography. In planar chromatography, stationary phase is present on a plane. Stationary phase is present on a plane, and the plane can be paper impregnated by a substance acting as a stationary phase. That means it is soaked in a substance acting as a stationary phase. Plane can be paper and it is soaked in a stationary phase. The paper's chromatography or a thin layer of substance acting as stationary phase spread on a glass, metal or plastic plate. This is thin layer chromatography. So we have to note down when we use paper, it's paper chromatography, and when we use the thin layer of glass material, it is thin layer chromatography. And what are the different application of chromatography? Chromatography technique is used for the separation of amino acid, protein and carbohydrate. And it is also used for the analysis of drugs, hormones and vitamin. Helpful for the quantitative and qualitative analysis of complex mixture, and the technique is also useful for the determination of molecular weight of protein. So these are, and other than this, it is very useful to find out the adulteration in food. And paper chromatography is a technique that involves placing a small dot or line of sample solution onto the strip of chromatography paper. The paper is placed in a jar containing shallow layer of solvent and sealed. As the solvent rises through the paper, it means the sample paper mixture. And here is the procedure. Just look at the picture. A small spot of sample is applied to the strip of chromatography and about 2 centimeters away base of the plate, and this sample is absorbed onto the paper and may from interaction with it. The paper is then dipped into the solvent such as ethanol or water. Taking care from the spot is about the surface of solvent and placed in seal. So just you look, two phases is present on chromatography. One is stationary phase and another is mobile phase, and how the experimental setup is. Simple chromatography, we have to put a spot on the above the baseline and allowed to pass calculating RF value. These 2 points are very important: distance sample travel and distance solvent traveled. Once again, please note down the important points included in the paper chromatography. Paper chromatography is a liquid partition. In paper chromatography, the end of the paper is dipped in solvent mixture consisting of aqueous and organic component. The solvent soak in paper by capillary action because of the fibrous nature of paper. The aqueous component of the solvent binds to the cellulose paper and thereby forms stationary phase in it. The organic component of binds continues migrating, thus forming the mobile phase. So these are the procedure which included in the paper chromatography, and we have to find out the RF value. RF value can be, it is very useful to help the identify the unknown. And how we can calculate the RF value? Distance traveled by the substance by distance traveled by